Hi everyone, and welcome to Chapter 28, Invertebrates, Part 1. So this chapter begins our study of animals, beginning with the invertebrates. Now, invertebrates are animals without rigid spinal columns or backbones. The invertebrates have dominated the animal landscape for millions of years. In fact, 97% of extant animals, that's living animals, 97% of the animals on the planet today are invertebrates. That includes insects, spiders, crabs and lobsters, worms, jellyfish, we call them sea jellies, clams and oysters and corals and sea sponges, all examples of invertebrates. So the invertebrates exhibit an enormous variety of cell and tissue adaptations for specific purposes, including outer cuticles for protection, spines and tiny harpoons for defense, toothy structures for feeding, and wings for flight. An exoskeleton can be adapted for movement or for the attachment of muscles, as in the case of clams and insects. And some invertebrates have secretory cells that can produce venom, mucus, or digestive enzymes. Our objectives for this first part of Chapter 28 explain the form and functions of sponges, identify the two general body forms of the Cnidaria, that C is silent, we say Cnidaria, describe the unique anatomical and morphological features of flatworms, describe the unique anatomical and morphological features of mollusks, describe the formation of the coelom, discuss the advantages of body segmentation, and describe the features of segmented worms, describe the structural organization of roundworms, and compare the life cycles of parasitic worms, and also describe the life cycle of the octopuses. Some people say octopi, and that's fine. Okay, so here we go. So the first phylum in our study of the invertebrates are the Porifera. Now, POR comes from that word that means pore. So you can imagine these are animals that are porous. That'll make more sense as we look at the description. Porifera are animals with pores, like sponges, sea sponges I'm talking about. They're the simplest invertebrates. They have no true tissues. The larvae have flagella, which means they are able to swim around. The adults, however, are sessile. Sessile means that they are fixed in a fixed position on the substrate, on the floor of the sea or on a rock. The larva can swim, however. Now, the general shape of the sponges are cylindrical. That's the most common phenotype. Pores facilitate the movement of water into and out of the sponge. And the pores are needed for feeding, excretion of waste materials, gas exchange as well. Now, sponges are filter feeders, as you might imagine. And the sponge's seal, just some of the morphological descriptives, the sponge's seal is a central cavity where water enters. And porocytes are cells within the walls of the sponge that filter things out, that capture food items, prey items. Porocyte, these are cyte cells within the organism, which is a porifera. Now the osculum is the opening at the top where water is expelled. So you have a tube-shaped sponge that is fixed to the ground. At the top there's an opening, so it's like a big long tube, and that opening is called the osculum. Now the mesohyal is a gelatinous substance between the layers of tissue, has no real tissues, but between the layers of structure, and that helps to maintain form. Amoebocytes, now these are cells that are amoeba-like. They're stem cells, and they reside in the mesohyle, and they give rise to oocytes. Now, oocytes, as you know by now from the first semester, are your egg cells or your ova. Oocytes are the cells that make ova, make eggs. Amoebocytes give rise. Now, remember, they're stem cells. So from amoebocytes are generated all of these other types of cells, like oocytes, chelinocytes. They make collagen, sclerocytes, choanocytes, which are flagellated. Now, spicules are made of silica. That's a glass-like material, or calcium carbonate. And spicules take the form of like little spikes or little spiked boxes or spiked balls. And they are used for protection. Obviously, they're made of a hard substance, so they're like they're sharp. So any predator that might come along that might bite into a sponge would get these prickly sensations in their mouths, and they would effectively leave the sponge alone. Reproduction for sponges is asexual via budding on the one hand, and there is a sexual phase of reproduction. Now, sponges are monoecious, and you know from earlier studies that monoecious means that the male and female gonads are in the same organism. There's no male sponge and female sponge. The sponge has both gonads in them. So they can self-fertilize on one hand, and then on the other hand, they can bud. So here's some examples of sponges. Now the sea sponges is what we're talking about. So here we're looking down on this series of sponges. They're fixed to the substrate, whatever that is, a rock down there. And you can see this central opening there. Now if we look at these sponges from the side, they look kind of like this. So you see these long tubes rising vertically. Now, some of you actually may use sponges, for instance, exfoliating when you take a bath or take a shower. Now, this is not a loofah. Loofah is a sponge-like structure, but that comes from a plant. But this is a natural sea sponge right there, which does the same thing. Okay, so looking at some sponge form or morphology, here's the adult sponge fixed to the substrate. There's the osculum, that large opening at the very top. And this large space inside is the spongiocele. Now, of course, this is in cross-section. Is the spongiocele, and that is where water is pumped into, from the outside, pumped into the spongiocele. And here are the chelinocytes, which are flagellated. That's what these little squiggly lines represent, flagella, that help to maintain the flow of water, the current in the spongiocele, but also those little flagella act to trap particles of food within the spongiocele, coming into the spongiocele from the outside water. Now over here are cell types. Here's your amoebocyte. It says delivers nutrients to cells, but also differentiates into other types of cells. So these amoeba type cells differentiate into chelinocytes. There's your oocyte, which is your egg cell, and the lophocyte and chelinocyte for making collagen. Chelinocyte for making collagen. Porocyte control. That's the cell here that's controlling the flow of water coming in. So these are long porocytes here. And sclerocyte down there, secreting silica spicules, those little sharp points. So if you go out into the ocean and you grab a sponge, you feel little prickly spicules. All right, let's go to the first video all about sponges from NG Science. See you on the other side. And a quick check. Which of the following statements about sponges is false? Your choices. Choanocytes have flagella that propel water through the body of the sponge. Pinacocytes can transform into any cell. Colonocytes secrete collagen. Porocytes control the flow of water into the spongiocele. And I thought there was another one. Okay, so the statement that's false is pinacocytes can transform into any cell type. Remember, that's the amoebocyte that can transform into any one of these cell types. Choanocytes have flagella. Colonocytes secrete collagen. There's collagen. And porocytes control the flow of water into the spongiocele. See the porocytes, those are the cells with the pores through which the water flows from the outside environment into the spongiocele. And that's why these animals are called sponges. By the way, yes, they are animals. Okay, so the next phylum in our survey of the invertebrates is the Cnidaria. The C is silent, Cnidaria. So the major cell type is the cnidocyte, which is a cell that stings. It's a stinging cell. That's where the phylum gets its name, from the cnidocyte. Okay, so now nematocysts are little organelles with harpoons on them. I'm telling you, these are extremely simple animals, but so sophisticated in what they do in their environment. The natural world, I always say the natural world never ceases to amaze. And this is one of those amazing things. So the harpoons are sharp barbs attached to a hollow thread. Now, check this out. It's amazing how this works. So, cnidocils are hair-like feeler cells, extensions sensitive to touch. Now, when cnidocils are stimulated, the cnidocytes, stinging cells, fire their nematocysts with their little harpoons. The barbs of the harpoons penetrate prey, and then the animal, the cnidarian, pumps toxins through the hollow thread into the prey, and that's how they subdue their prey. Isn't it amazing? And these are simple little animals. So corals is an example of a cnidarian. Corals have these little stinging cells. Now, morphologically, the polyp is sessile. Now, sessile means it's fixed to the substrate, fixed to the bottom of the ocean, or fixed to a rock, or whatever. Okay, it doesn't move. Right? It's like a tree. It doesn't move anywhere. So that's what we mean by sessile. That the form of the cnidarian is referred to as the polyp. Now, the medusa is the form of the cnidarian that's free swimming. And so what happens is when new cnidarians, and think about coral, this is interesting. When new corals are born, they're free swimming and they're able to swim around. And then they'll find a place and they'll settle into the substrate or settle on a rock, and they'll affix themselves to the substrate, and then they'll grow into adults. And they never move from there. Once they attach themselves to the substrate, they never move, and they grow into adults, and now you have a new coral, a new what you know as coral. Interesting. So we call this process or this program dimorphic, right? Two forms, a sessile form, the polyp, and a free swimming form, the medusa. Now, cnidarians include corals, like the Great Barrier Reef, and marine jellies. You call them, well, lay people call them jellyfish, but they're sea jellies or marine jellies. They're not fish, so this is really a misnomer. And hydras. You might have, okay, so there is an example of a coral reef. And so these are the polyps, the coral polyps that are fixed to the substrate. There's a jellyfish or a sea jelly or a marine jelly. Of course, you're familiar with those. And especially if you've been stung by one, right? You go down to the beach and you go swimming in the ocean, and there's certain times of year where the jellies are just swarming, and you might get stung by them. And, and then 1,000 people will tell you 1,000 different remedies to put on your wounds to stop the stinging. And I won't go into some of those because some of those are pretty bizarre. And that's a hydra. Now, hydra is a little cnidarian, little freshwater cnidarian. And this is something you may have played with in high school biology. In not you guys, but 7th grade life science, or in regular biology class, you might play around with hydras. They're kind of neat. Okay, so here is a cnidocyte. This illustration is from your text. So this is the cnidocyte, and there's the little hair-like projection, the cnidocyl. And remember the opening at the top, just like with sponges, called an operculum. This one happens to be hinged. And so when the cnidocyls get stimulated, the little door pops open, and the nematocyst fires out. It's got the barb attached to a little hollow thread. There's the barb. There's a little harpoon. It sticks into a prey item. And there's the little thread, which is hollow. And now the cnidocyte can pump its toxin through the little tube and into the prey, or into you if you happen to be unlucky and get stung by a jellyfish. And you know, if that's ever happened to you, you know how that feels. Okay, morphologically, these are dimorphic animals. So there's the medusa. Now that kind of looks like a jellyfish, right? Or a sea jelly, as you know them. And so that's the medusa form. And here's the polyp form. The basal disc here is fixed to the substrate. The gastrovascular cavity, they don't have a stomach, they don't have a digestive system as such. There's the cavity. And we have ectoderm, and the mesoglia is this fluid between layers of ectoderm and endoderm. Okay, there's no mesoderm here. Tentacles here, tentacles down there. That's the operculum, which is the mouth. There's the operculum on the bottom side of the medusa. This, by the way, is both the mouth and the anus. So this is where food comes in and waste products leave. All right, now within the phylum Cnidaria, we have a class called the Scyphozoa. Now, Scyphozoa means cup or cup-shaped animal. And this includes your jellyfish or sea jellies, and there's about 200 species of them. Now, the medusa is the prominent life cycle stage for the sea jelly, as you know. The medusa is free swimming, and these are solitary animals and only come together when mating is necessary. Otherwise, they kind of like to be alone. So when you're out in the ocean, down at the Outer Banks or whatever, and you get a massive influx of sea jellies, of jellyfish, that's a mating thing, right? It's a mating collection going on, a mating event. Okay, so the mouth is located on the underside, as we saw from the illustration. Tentacles contain the nematocysts, and these are carnivorous animals. They eat other animals. The nervous system is very simple. Consists of a ring of nerves, or a nerve ring, and then a nerve net that kind of flows through the rest of the animal. We say innervates the rest of the animal. So there's kind of a central ring. We'll see it in the illustration, and then a net that flows from there, that's attached to there or extends from there. Now, muscles provide force for swimming and for drawing in food. And you've seen, at least maybe on Discovery Channel or something, how a jellyfish, how it sort of undulates to swim. Well, of course, it has to have muscles. Muscle means movement. So on the reproduction side, sexual reproduction, these animals are dioecious. So there are male sea jellies and female sea jellies. So fertilization, though, occurs externally. So the females release the eggs, the males release the sperm swim in the water to fertilize the eggs. You got zygotes, and then you have new sea jellies. The gametes themselves are expelled through the mouth because there's only one opening. Now, larvae settle on the substrate and form a polyp. Now, remember, the polyp is fixed. So they're dimorphic, okay? Cnidarians are dimorphic. So here's the polyp form. So the larva, which swims, it's going to swim around, and then eventually it's going to settle on the substrate, and it's going to attach itself and form a polyp, and the polyp is going to get larger, and then the polyp is going to bud, and then new medusa are released, and those new medusa are baby jellyfish. So here we see a medusa, of course, and the mouth would be down here. There, of course, we don't see it here, and there's some of those tentacles. And so here we see the medusa. This is the predominant form for the sea jelly. There's the mouth, which is also the anus. What else? The nematocyst-bearing tentacles are here, so you see them here kind of flowing backwards. Gastrovascular cavity, so that's where food comes in. And we have epidermis there, and mesoglia to fill the cavity, fill the space. And gastrodermis. So this is the layer of tissue that lines the gastrovascular cavity. So we have epidermis, gastrodermis, and the mesoglia fills the space between. Mesoglia helps to give the animal form. So only two tissue layers here: epidermis, the outer dermis, and the gastrodermis. Okay, pretty cool animal. So the life cycle of the sea jelly, I don't want you to so much sit down and memorize, because I'm going to show you lots of life cycles, but just to give you an idea of what's going on in the natural world. So here's the medusa. The medusa makes gametes, sperm and egg. Sperm fertilizes the egg or ovum to make the zygote. The zygote turns into the larva. The larva is free swimming, but once it finds its spot, it fixes itself, becomes sessile, grows into a polyp. The polyp gets larger, it buds, and when it buds, new medusas. So medusa makes the gametes, fertilization occurs to make the zygote. The zygote hatches, if you will, into the larva. The larva grows into the polyp, which is fixed. The budding polyp releases new medusa, and now you have new baby jellyfish. All right, next video all about Cnidarians, again from NG Science. See you on the other side. By the way, all of those videos are, if you just look in your course menu to the left, go down to whatever module, and it will say embedded videos for chapter 33, 34, 2, 5, or whatever. It'll say embedded videos, and that's where you'll find all these videos. And they're just for your edification, just for your knowledge. If you're more interested in things, I mean, because some of these sections will have a lot of videos. I just find tons of stuff, and I just put it out there. Okay, quick check. Which of the following statements about Cnidarians is false? Sea jellies are monoecious. The medusa is most prominent in the life cycle of sea jellies. Nematocysts are located on tentacles. Sea jellies are carnivorous. I don't think I have another one. So if we go through them, sea jellies are dioecious. Remember, you have male sea jellies, male jellyfish, and female jellyfish, okay? So that's definitely false. And the last one there was nematocysts fire when cnidacils are stimulated. So the Great Barrier Reef is made up of cnidarians. So as we've talked about, and as I just mentioned a little while ago, sea jellies are dioecious. There are male jellies and female jellies. The medusa is the most prominent in the life cycle of jellies. Remember, the polyp is just very temporary until it can grow big enough to release new medusas, which are what you know as a jellyfish. Now, nematocysts are located on tentacles. Those are the little stinging, the little barbed stingers. Sea jellies are carnivorous, of course. All cnidarians are carnivorous. Nematocysts fire when cnidacils are stimulated. Remember, cnidacils are those little hairs that stick up, and yes, when they get stimulated, boom, little harpoons fire off. And the Great Barrier Reef down in Australia, Barrier Reef, it's a coral reef. Corals are cnidarians, so the Great Barrier Reef is certainly made up of cnidarians. Preguntas? Any questions?